Hi, I'm Elvin. I'm going to present you my third year project, which is Healthy Aging Inside the Brain. This project is under supervision of Dr. Fumi Coston, and this project is about visualizations of the physical changes in the human brain throughout the aging process. In this screencast, I'm going to talk about the backgrounds about brain aging, how I produce brain images at different ages, and the synchronized image viewer that I created for better visual experience. Aging plays an important role in organ degeneration. When we age, our chronological age is often different from our biological age. For example, we could be officially 50 years old, but the age of brain could be 70 years old. When our biological age increases, our cognitive ability declines in this process. Aging is the main factors of age-related diseases such as Alzheimer's disease, Parkinson's disease, and dementia. If we can stop this process at the early stage, we can prevent from the age-related diseases. In this project, we focus on the physical changes in our brain throughout the aging process. Our brain volume reduces at the rate of 5% every 10 years after 40 years old. After 70 years old, the rate of decline increases. The affected brain tissues in this process are gray matter, white matter, and cerebrospinal fluid. As you can see from the diagram, the center ventricle size in the 18 years old brain has smaller empty hole than the 80 years old brain. This is due to the fact of the younger brain is filled with more white matter cortex, so it looks less empty holes than the older brain. My project is to visualize the structural effects of aging on the brain so it can assist the diagnosis tool for age-related disease. The main achievement in this project is to develop region algorithm for the brain data generation and then testing the generated data with the real MRI scan data, and also created a synchronized image viewer. I used the brain data provided by Riken Institute, which is a set of brain data from the MRI scan of 55 Japanese male scientists in the interval of 2 millimeters every single slice. This is the examples of some slices of the provided brain data. However, this data consists of only one cross-sectional plane, which is horizontal plane. I preprocess the data to get another two cross-sections which are coronal and sagittal planes, in order to deliver the three dimensions visual views of the brain. This is the preprocessed data. The first one is horizontal plane, the second one is coronal plane, whereas the last one is sagittal plane. Next, I developed an algorithm that can generate brain images at different ages using the provided Riken files. This algorithm is inspired from the mathematical morphological operation. It has a small set of pixels that is grouped in a specific pattern way that called structure elements that can probe through the image pixel and then perform enlargement or shrinking on the particular regions on the brain images. This is the illustrations of how the region processing works. As you can see, the small set of pixels change the border between two big sets of pixels into its corresponding pixels value from the value of 2 to value of 1. This is the generated result. The first row is the 40 years old brain, whereas the second row is the 80 years old brain. I evaluate the accuracy of the algorithm with the real MRI scan. I use OSIS1 dataset as my testing data. The evaluation method that I created is to measure the ratios of the center ventricle size to the size of the whole skull. I calculate the distance between the points as shown in the diagram using screen ruler and then measure the ratio of each stage. This is the ratio differences between every 5 years from age 20 to age 90. You can see from this horizontal plane the volumetric changes in the generated dataset and the testing dataset is not much different. Next, I perform the same thing on the coronal plane, but the points locations are different. This is the result I got from the coronal plane. The another plane is sagittal plane. I also perform the same thing but with different points positions. This is the result for the sagittal plane. The next thing I do is I created a friendly user interface that provides synchronized image viewers. This is the created web applications. Let's put in the age 80 years old and 80 years old. You can see both figures change their corresponding images when click on the different areas on the brand images. These features helps the user to see the differences between two brand images at two different ages when they want to test out the generated data. All the brand images at different ages are stored in local server. I developed these features in JavaScript, and the program reads all the local files and presents it to the user. As a conclusion, this screencast presents the developed algorithm for the brand data generation of different ages and delivered satisfactory results in the comparisons of the algorithm with the real MRI data. In the end, the artifact with the synchronized image viewers features are shown in the video clip. Thank you very much for the listening.